All right, so continuing on here with chapter 6, we're going to talk about oxygen requirements. So, the bacteria that are sort of kind of like humans, okay, bacteria that absolutely must have oxygen in order to survive are called obligate aerobes. Now, there's bacteria that can actually use oxygen when it's present, but they don't require oxygen in order to survive. And they are called facultative anaerobes. So again, we have those terms obligate and facultative, and we've already had aerobic and anaerobic. So now we're kind of putting those terms together. And of course, you want to make sure you know all these for the test. I can guarantee that all these will be on there. So now there's actually lots of bacteria that live in oxygen-free environments. So they actually are damaged or killed by oxygen. And they are called obligate anaerobes. Another group is bacteria that don't use oxygen at all and are not harmed by it. So they basically just don't care about oxygen at all. They don't use it. It doesn't hurt them. It's just a non-issue for this group. And they're called the aerotolerant anaerobes. And then finally, we have microaerophiles. These are bacteria that use oxygen, but at very low concentrations. So treat these as vocabulary words. You want to make sure you know the definitions of all of these. Now, not only do you need to know the definitions based off of oxygen requirements, you also need to be able to identify the bacteria based off of their growth in a tube. And that's what this picture is showing you. So basically the first thing is the definition, and then the second are the actual tubes. Now, what I will do on the exam is I will have actual pictures of these tubes here, okay? And then you need to identify which type of bacteria is shown in that tube. It'll still be multiple choice, but you do need to be able to identify these. And we're actually going to do a lab about this as well. So a very important topic. Now, this picture is on page 155 of your book. All right. So obligate aerobes, the bacteria that absolutely require oxygen, the growth is going to be seen near the top of the growth medium. So basically this yellow stuff, think about it like Jello. It kind of has the consistency of Jello, okay? So it's not a liquid, it's pretty much a solid, but it's like Jello, okay? So this would be where the oxygen is, right? So this is where the air in the tube is. So the pink dots represent the bacterial growth. So you can see the obligate aerobes, they're going to be like towards the top because that's where the air is, okay? Facultative anaerobes, they're going to be all over, okay? But especially at the top, because facultative anaerobes, they will use oxygen when it's there, but they don't require it. So they're going to kind of grow throughout the whole tube. Now, the obligate anaerobes are the opposite of obligate aerobes. So they're going to be all the way at the bottom, as far away from the oxygen as they can get. Okay? The aerotolerant anaerobes are very similar to facultative anaerobes. The difference is they don't care about oxygen at all. So there's not going to be a whole lot up at the top. They're basically going to be spread out throughout the whole tube. And then finally, the microaerophiles, they're right in the middle because they don't want a lot of oxygen, but they want some of it. So they're kind of right here in the middle, kind of like Goldilocks and the Three Bears. Not too much, not too little, just right in the middle for them. Okay, so again, make sure you know the definitions of all 5 of the oxygen requirements, and make sure you can identify the bacteria based off of these tubes. All right, so more about oxygen. Now, of course, oxygen is O2. So, that's what we breathe. That's what the bacteria is going to, take in and all that kind of stuff. Well, there is such a thing as singlet oxygen, which is basically like kind of like an O instead of an O2. And that is highly reactive with organic molecules. It's going to want to bind to pretty much anything it can bind. Superoxide free radicals steal electrons from neighboring molecules, and that's basically oxidation. Like, we get the word oxidation from the word oxygen. Oxygen is the reason that we age. Oxygen is the reason that fires happen. Oxygen is the reason that rust forms on metal. So oxygen, we definitely need it, but it can also cause some damage. It can cause some problems. I know you've all heard of peroxide before, and peroxide is probably the most important one from this list. Most likely peroxide will be the only one that shows up on the test, okay? Because peroxide is used as a disinfectant, and it is an oxygen derivative. A peroxide is H2O2, which is actually derived from oxygen and water. And we'll talk a little more about peroxide in chapter 7, as it is also a disinfectant. I'm sure you've all heard of ozone before. The ozone layer. It's what helps to keep the harmful rays of the sun from getting to the earth. But as a molecule, ozone is O3, so it's actually 3 O's instead of 2. And it can cause DNA mutations and other type things. And then hydroxyl radical, which is an OH, is another very reactive form of oxygen. Now, if you want to know more about these, you can absolutely read more about it in the book. The book will tell you more about like the chemistry, behavior, and that kind of stuff. But like I said, the only one you really need to know for the test is peroxide. Okay, growing bacterial cultures. So this is something that we may, I hope, be able to actually do. Okay, so this is a lab. This is actually several labs that we would do where we're going to try to grow bacteria in petri dishes. And in a few weeks, we're going to try to see if we can get you guys to be able to do this at home. So that's going to be a lab. It'll hopefully be really fun, really interesting, and we will eventually get to that. So the medium or media, that's the stuff that the bacteria grows on. And a lot of times it's either a liquid broth or it's a solid kind of like jello, like I said before. Now, when that bacteria starts growing on the medium, it's called a culture. And a term that not a lot of people would use, but the term for when the bacteria is actually introduced to the medium, it's called an inoculum. But that's a very technical term. Most people are just going to use the term culture and just say, hey, this is a culture of bacteria. So as far as the media goes, again, that's kind of like either the broth or the jello-like substance. It has nutrients, it has water, it's pH balanced, and it's sterile. You have to make sure there's no other bacteria in there, or else that's going to affect the growth of your bacteria. And then there are other types of media that are used to grow specific types of bacteria. We do have a lab where we do that sort of kind of, but that's not going to be a lab that you're going to be able to do at home, so we do a virtual version of it. Okay, so I keep saying like you know, it's kind of like Jello. It's kind of like Jello. Okay. Well, this is where we talk about the agar that's in the media. Okay. So agar is a polysaccharide that's derived from algae that acts as a solidifier. So you know how like when you make Jello and it's a liquid, right? You'd start off with like the boiling water and you mix it and it's a liquid, and then you pour it into whatever you pour it into, you put it in the fridge and it solidifies, right? It like gets harder, right? That's what agar does for the media. So the media is going to have things like, you know, again, water, carbohydrates, other things that keep it pH balanced, okay? And it needs a solidifier. It needs to kind of become more firm. And that's what the agar does. Very, very important in microbiology. The bacteria don't eat it. You know, it's pretty cheap and easy to come by, and it's stable in different temperatures, except it does not like colder temperatures, but it definitely is fine. Refrigerated, room temperature, hotter temperatures, all that stuff, very important. So if the media is in a test tube, it's called a slant. And then a petri dish, once the petri dish is inoculated and becomes a culture, oftentimes the petri dish is called a plate, but a lot of times people will just call it the petri dish or the culture. And petri dish, by the way, the P should always be capitalized because it is named after the person who invented it. So again, my hope is that we will be able to have you guys working with some petri dishes, be able to do a nice, fun, hands-on lab that you guys can do at home. So hopefully that'll be fun. All right, so this picture, not super duper important, just to kind of talk to you a little bit about this kind of stuff. This is on page 162, and this is just talking about the different types of media, okay? So the media that we pretty much use in our lab is called complex media. That's used to grow just the regular, normal, everyday chemoheterotrophic bacteria, okay? Reducing media is used for obligate anaerobes. Selective types of media encourage desired bacteria to grow. Differential media is for the differentiation of bacterial colonies. And enrichment is like a super version of selective, okay? If I were to ask you a question on the test about media, it would probably just be, you should know that we use complex media in our lab. Okay, so like I said, this is not a big deal for us, but it's just a topic that we want to cover and at least mention. All right, so for this one, I'm trying to think for the lab that we're going to try to get you guys to do at home, you're actually not going to be doing what's called the plate streaking method, okay? Because you're not going to have access to a Bunsen burner, and we don't want to have anybody doing anything with fire at home or anything like that. So you're not going to actually do the plate streaking method for, you know, the lab that you actually do at home, but it's something you need to know about, and it is something that may come up in other labs, okay? So basically, you kind of follow the steps, 1, 2, 3, okay? You get some bacteria from a broth or from some sort of stock bottle or whatever, and you do your first streak up at the top. Then you have to heat the inoculating loop to kill any of the bacteria that's on there, and then you do your second streak, heat it again, do the third streak, and what you're trying to do is you're trying to get individual little colonies of bacteria. So you see there's a colony, there's a colony, there's a colony, and then you see some of these are yellow. So you have a lot of red colonies, and then you have some yellow colonies. Those are probably 2 different types of bacteria. And this is the method that's used to obtain, you know, a pure sample of just one species. All right, we're going to go ahead and end the video here, and we'll pick up with the rest of chapter 6.